Bismillahirrahmanirrahim. Today we shall talk about bacteria. This is a review slide. There's a lot on this slide, but this is everything that we have already covered. So I'm just going to go over this because review is where you actually learn the material. So as we talked about before, we have the 6 kingdoms, and 2 of the kingdoms are dedicated to bacteria. And these are the kingdom Archaebacteria and the kingdom Eubacteria, and these are the prokaryotic kingdoms. The other kingdom Protista, kingdom Fungi, kingdom Plantae, Animalia, kingdom Protist, fungi, plants, and animals, those are all eukaryotic kingdoms. So anyway, among the prokaryotic kingdoms, the Archaea and Eubacteria, we previously talked about the Archaebacteria. These are the anaerobic bacteria which live in the extremes of environments, reminiscent of what the Earth was like in times of old. So these we classified into 3 classes, and these are 3 extreme environments. So the first category of these Archaebacteria were the methane-producing bacteria that lived in oxygen poor. In fact, all of these bacteria don't like oxygen, not that because there was not much oxygen on the planet in the beginning of its life, as scientists suggest to us. So the methane-producing, these are in the marshes and in the tummy of the cows and in sewage plants. That's the first category. And then there's extremely salty water bacteria, and you can find them. And then they're in acidic water, such as in ocean floors. The kingdom Eubacteria can similarly be divided into 3 categories, but of different sorts. These include the heterotrophs, so the parasites that what we like to think typically as a bacteria. And these feed off other living things. And the saprophytes, they feed on dead organisms or detritivores, like for example, and they feed on dead organisms or organic waste. So these are heterotrophs, and then there are photosynthetic autotrophs bacteria, and these are the first oxygen-producing living things in the planet, we understand. So these are photosynthetic autotrophs. They have chlorophyll and they appear green, and the example of this is the photosynthetic cyanobacteria. Now, photosynthesis is the production of energy-rich compounds such as sugars using the energy that's in light. So photosynthesis traps light energy and converts that or saves that light energy in the form of organic molecules such as glucose. The other way of producing energy-rich compound is chemosynthesis. Now these are autotrophs. These are also eubacteria. They are autotrophs. That means they are independent. They make their own energy, but they make their energy by chemosynthesis. So what they do is they take they're not light dependent. They use for example, you can find this in hydrothermal vents in the ocean floor. They take carbon dioxide and ambient water and hydrogen sulfide and oxygen, and they produce out of it, voila, carbohydrates like glucose. And the byproducts in this case are not oxygen. The byproducts are sulfuric acid and things. So there are other ways of producing glucose other than photosynthesis, right? And using other molecules. Now, to make glucose, you're going to have some carbon source, right? In this case, carbon dioxide is still the source, and you have to have some oxygen source, and that's usually water and oxygen. So as long as you have the ingredients available, you can produce glucose molecules from by different methods. So again, in eubacteria, right, they're heterotrophs, the typical parasites or saprophytes, and that we typically think of bacteria as. And then they're the photosynthetic autotroph bacteria and the chemosynthetic autotrophic bacteria. Chemosynthetic autotrophic bacteria are also important, as we'll see, because they also fix nitrogen, and that's very important, which we'll talk about in a little bit. So this was all a review of what we did before. This slide is an example of chemosynthesis. So in this case, carbon dioxide plus water plus hydrogen sulfide produces sugar and sulfur compounds at the bottom of the sea floor. For example, over here in mussels, apparently that's what they do over here. So as long as you have, in this case, hydrogen sulfide is one of the ingredients here. As long as you have the requisite ingredients, you're able to produce sugar molecules, which are used then for energy. So These are two types of autotrophs. This is a formal discussion of bacteria for us today. So bacteria are prokaryotes, and prokaryotes, as you recall, they do not have membrane bound organelles. They do not have intracellular membrane bound organelles, such as the nucleus, such as the mitochondria, such as vacuoles, such as endoplasmic reticulum. None of those structures will be found in bacteria. Bacteria are small. Typically, they're one-tenth the size of a eukaryotic cell. Sometimes they can be bigger, sometimes larger. So, but they're typically much smaller than eukaryotic cells. Here's a bacteria. Now, you can't have a cell without having a first cell membrane. The cell membrane is the boundary of the cell. It's like kind of not having a house without. It's trying to have a house without four walls. How are you going to have a house without four walls, right? You know, that's not a house. It's like, you know, I don't know what that's, a gazebo or something. But so you have to have a cell membrane. So all living things have cell membrane. Bacteria have cell membrane. Amoebas, protists have cell membrane. Plant cells have cell membrane. Your cells have cell membrane. So everybody has cell membrane. That's nothing special, so to speak. And that's very special, so to speak, in a sense, because everybody has cell membrane. And that's not special because everybody has cell membrane. So either way you look at, bacteria and all living creatures have cell membranes. But what's special about bacteria is, in addition to the cell membrane, they have cell wall. So they have a cell membrane. It's the plasma membrane, and outside the plasma membrane, they have a cell wall. The reason is because, imagine, these dudes have been around for over 3.4 billion years old. I mean, they have to be pretty sturdy. That little cell membrane, the plasma membrane, is not going to carry you for 3.4 billion years because they lived in such extreme environments. So they have an additional protective layer to protect themselves from many different things, and primarily what we'll see, water loss. Okay, so these cells, all cells, are very sensitive to water loss, either water loss or water gain. So plasma membrane is relatively permeable to water, and the water shifts can occur across plasma membrane readily, depending upon the tonicity of the environment, depending on how much solutes are in the environment relative to the inside of the cell. So cell wall is a very important component of the bacteria, because bacteria are unicellular living things. All they got for protection is all they're carrying on their back, so to speak. So in addition to the plasma membrane, they have a cell wall. So cell wall prevents osmosis from bursting the cell, okay? Because the intracellular, they have a lot of solutes inside. The water can just gush in and burst the cell. So cell wall is very important in not imploding this bacteria, basically. So that's a cell wall. So in addition to the plasma membrane, all bacteria have cell wall. Easy peasy? That's not that difficult, right? Now, bacteria also have chromosome. They have a single circular DNA that contains most of their genes. So bacterial DNA, see, our chromosomes, the 23 pairs that we have, each chromosome is like long strand of DNA. And then we have like 23 pairs of strands. So they're like 46 strands bundled up inside the nucleus, right? So each of them is like a thread, a strand. But the bacterial DNA is like a thread tied to each end, like it does B, you know? So bacterial DNA is a circular DNA. And a single circular DNA, it contains most of the genes. Well, what do you mean most of the genes? Where are the rest of the genes? Well, most bacteria have tiny circular DNA, like tiny baby Tusbies, you know, that contain some of the other genes. They're called plasmids, okay? So the watery, gushy stuff here, where everything swims inside the bacteria, that's the cytoplasm. Ribosomes are there. They do protein synthesis like anywhere else. That's their job. So all bacteria have cell membrane. On outside of cell membrane, they have cell wall, and they have a single circular DNA that contains most of the genes and cytoplasm and ribosomes. Now, all bacteria have cell membrane. All bacteria have a cell wall. Some bacteria, these are like the commando bacteria, like they're like super tough, like, you know, super tough, super duper tough. In addition to the cell wall, that's not enough for them. See, they go out shop and they get themselves a capsule. So they have plasma membrane, cell wall, and a capsule on top of a cell wall. And this capsule has a different biochemical makeup. Cell membrane is plasma membrane is a phospholipid bilayer, right? Phospholipid bilayer, and cell wall is a proteoglycan. It's a proteins plus sugary material. So the structure of cell membrane is a phospholipid bilayer. The structure of cell wall is different than the cell membrane. It's not a lipid thing. It's a it's a protein and a sugar. We should study that actually in the future in a little bit detail. The capsule altogether is also very different. Now, not all bacteria have capsule. Okay, it's like a gelatinous material. So if you can think about a bacteria like a, French fry, the bacteria with the cell membrane and wall would be like the French fry. And if you were to dip it into ketchup, now you get a ketchup on top of this a French fry, and that would be the capsule. Now the capsule is very important because bacteria that have a capsule, they tend to be more pathogenic. They tend to be pathogenic means they cause disease. Most bacteria are not pathogenic, but many bacteria obviously are. So bacteria that have capsule, they tend to be more pathogenic. I can tell you that from personal experience. I saw a patient die in front of my eyes within several hours because of Pseudomonas aeruginosa bacteria. Pseudomonas aeruginosa. I remember that because that bacteria has a capsule. It's a dangerous bacteria, and I still remember this day because we couldn't do much for that patient and the infection. The capsule of the bacteria is an additional structure that some bacteria have. So a bacteria can have three layers potentially. It can have a membrane, so a plasma membrane. On top of the plasma membrane, cell wall. On top of the cell wall, some bacteria have an additional capsule, which can make them even more pathogenic. Now, in addition, this is a different cartoon picture. In this picture, the green over here is the plasma membrane, and then outside the yellow, I believe here is the cell wall, and outside the red, I believe here is the capsule that some bacteria have. Now, this bacteria, this cartoon picture shows some other, decorations, if you like to, of special features that the bacteria can have. They go shop and get more stuff. So many bacteria have structures called plasmids. Now, plasmids are like small circular DNA. The main DNA material, the DNA is long circular DNA of the bacteria. That's this big thing over here. It's like a big mush nucleoid circular DNA. And the circular DNA in the bacteria is not like all over. It's like a little hairy thing clumped up. And the area that's called the nucleus. It's not a nucleus, not surrounded by any membrane or anything. But the area where there's most of the circular DNA, that's called the nucleoid area. Anyways, so that's the major circular DNA. But in addition to that, so many bacteria have small circular DNA. They're called plasmids. That's like over here and here. These are like small baby tasbihs, like if you like to think about this, So plasmids they contain a few genes. And a bacteria may have one or more plasmids, and they contain few genes. Probably the genes that the bacteria uses a lot. See, and this is very important. But this is also frequently how one bacteria, if it gets resistance to a certain antibiotic, for example, this is how they can exchange. This is how they can exchange genes. So bacteria can exchange genes with other bacteria using these plasmids. Now, when bacteria share their genome, they don't share the whole caboodle, the big gigantic nuclear circular DNA. They don't share that genome. What they really share is the little small circular plasmids, and we shall see that as well. So many bacteria have plasmids. Okay, and we use that to advantage as well for biomedical reasons. Okay. Some bacteria also have these structures called pili. They're like little evaginations of the plasma membrane. They go out like this, like little mini antennas. They usually, bacteria that have pili, they have multiple pili typically, okay? And the pili are very important. As you can see, they're like, kind of like tunneling out, right? As you can imagine, if there's another bacteria, if they connect to another bacteria, there's like a mini tunnel over there, right? And then things can go from one area to another. So pili can sometimes is used to exchange the genetic material between bacteria. So that's pili. That's plasmid ribosomes we saw. And this thing over here, this tail-like thing, is a flagellum. Like the sperm has a flagellum, and some bacteria have a flagellum also. Some bacteria have one flagellum, some have two flagellum, and some can have multiple flagellum. And this allows the bacteria mobility. It gives bacteria a certain degree of mobility, right? How does it know where to go, you say? It doesn't have any eyes, right? Well, that's a very interesting question, isn't it? Now, if you're curious, how does it know where to go? Well, it tends to go where by chemical attraction, you know, chemostasis. That's called chemostasis, chemical attractions. Anyways, we should hopefully someday study that. Well, so plasma membrane, so the cell wall, and this is a capsule. This is cytoplasm. Ribosomes, this is the pili. Nuclear circular DNA, and this is the bacterial flagellum. This flagellum, the structure of the flagellum is very similar to the same flagellum that you find anywhere else, as also in the male gametes of the sperms. So we talked about the bacterial structures. So the structures that are always present in bacteria include the main chromosome, the DNA, and ribosomes, the cytoplasm, the cell wall. All bacteria have cell wall, and all bacteria have cell membrane. Some structures that are found in some bacteria, but not all, include the capsule, the flagella, plasmids, and the pili that we have discussed previously. Now, bacteria are of particular interest to us, of course, because, one, they cause us a lot of trouble. They cause strep throat, they cause ear infections, they cause endocarditis, they cause appendicitis, they cause all kinds of stuff, right? They can cause pneumonia, meningitis. So bacteria cause us particularly lots of trouble. So we want to study them. We want to classify them. We want to know which ones can be killed with which antibiotic, because some antibiotics work for certain bacteria and they don't work for other bacteria, and then you have to use other antibiotic for that particular bacteria. So we are very curiously interested in classifying bacteria so we can better know our enemies, so to speak, and we have to know we have the right weapon for the right enemy, so to speak, right? And leave the ones that are harmless alone. So identifying bacteria. So one thing we can do is we can color bacteria. These are the red snappers. I believe this is a tuberculosis bacteria here you see over here, sometimes known as the red snappers. So anyway, so identifying bacteria. So it's a very fascinating thing. You have the ink that's in your pen, like an ink pen. You can use the same ink pen to just color anything and everything, right? So let's say if you have a slide and you just put any ink, any color, it's going to color any and everything, including bacteria if it's there, right? So that's the concept. That's how these stains evolve, because if people try to see if different bacteria stain differently. So one of the very common ways of identifying bacteria is just throwing different colors at them and see if they take particular color. In this picture, for example, there are two different types of bacteria. This is called Gram staining, and named after this gentleman Gram, who presumably invented this. Okay, now Gram staining is basically what you do, sequential coloring. It's like first, okay, you assume we have a patient specimen. Okay, the dudes do that. They cough up some stuff, and you want to see what kind of bacteria they got in the sputum, because they got bad pneumonia or something like that. So you put that in their sputum, their specimen, in, right? They take a little bit of that specimen and put it in a slide, right? For example, Okay, you put the little specimen of interest on the slide, and then you sequentially put different colors. First, you put, like, I don't know, I'm just saying. First, you put red color, and then you give it time, for the bacteria to take up the red color. Just like if you want to give a particular handkerchief some time to see if it takes the red color or not. So you put the red color on the slide, so let it sit there, and then you wash the excess red color, because otherwise you have red color everywhere. So you wash the red color, and whatever the bacteria take up will remain, right? Because it's hard to wash up stains, right? If you ever get up like a ketchup stain on you, or like some other stain, and you think you keep washing, it doesn't go away, because some stuff just take up that stain. I'm telling you. What is your clothing? Your clothing, it's oftentimes organic material. Organic material tends to take up stains. So anyway, so you take the little sputum, you take a sample, you put it on a slide, you throw some red dye on it, and then you give it some time for the bacteria, which, if they want to, absorb the red dye, and then you wash the excess red dye, and then you put some blue dye, if you happen to have some with your blue dye in there, and wait for some bacteria to take if they want to take it up, and then you wash up the excess bacteria, and then you look under the microscope, and then you notice something curiously. There's certain bacteria, they take up the red stain and they don't take up the blue stain, and there's some bacteria that take up the blue stain and they don't take up the red stain. That's kind of fascinating, right? And this is a consistent finding. So that's very interesting. And then you say, okay, then there are two types of bacteria. Those bacteria that take up the red stain, and those bacteria that they take up the blue stain, right? And then there will be bacteria that they don't take up any stains. Then you have to go look for more colors, don't you? That's exactly what you have to do, because if you go to microbiology, they don't just use gram stain to identify bacteria. They have a bunch of other stains as well. But this is the most common stain that's used that helps us discriminate between the common infectious bacteria. In other words, this is so useful, because it's easily able to tell us if, for example, if somebody got strep bacteria or not, because strep is one of those bacteria that takes up these stains. So gram stain divides the bacteria of the world into basically two parts, and there's a third category. The two parts are those that take up the red stain and those that take up the blue stain. The third category is, in fact, the bacteria that don't take up any of these stains. They're blue. Then you have to go look for other stains, right? It's McConkey stain and stuff like that. Okay, so, but gram staining divides the bacteria into two different categories. Gram-positive bacteria, they're purple looking, they're blue looking, and the gram-negative bacteria. What color would that be? That's like negative colors, like blood, red color. So gram-negative is red color. Gram-positive bacteria are bluish in color. Now, what part of the bacteria takes up this color? Is it the nucleus? No, bacteria don't have a nucleus. Is the ribosome? No, they're too tiny. Is it the endoplasmic reticulum? No, bacteria don't have endoplasmic reticulum. Silly. It is the cell wall. The cell wall, because cell wall is a pretty sturdy structure. It's kind of like your clothing type of sturdy structure, right? So the dye that's taken up, in fact, is taken up by the cell wall. And what type of a dye it takes really depends on what kind of a cell wall it's got. So cell wall is made of interlocking sugars and amino acids. I already suggested the cell wall is not like the plasma membrane. It's not made of a lipid bilayer. It has nothing to do, not lipids. It's the other important stuff, that is sugars and proteins. The cell wall are made of sugars and proteins. They're pretty interesting, complicated structures. So anyways, the staining process is staining the cell wall. This actual cell wall takes up the stain. So we can classify bacteria broadly as gram-positive and gram-negative bacteria, and this is a very useful classification. So here, in fact, you put the blue-violet application, and you put application of iodine, and your alcohol wash, and you put the application of saffron, which is red color. In fact, you start with the blue first, and then you do red color. This is just to help it stain better. So that's easy peasy. That's how it works. So gram staining bacteria can identify bacteria into two different categories. For example, the Streptococcus bacteria is a gram-positive bacteria, right? So if somebody's got a bad sore throat and somebody does a gram stain, and they see something like this, they don't got strep. You don't know what they got, but it ain't strep, right? So, and then that tells you, oh, we better be careful. Make sure we give the right antibiotic to this person. Otherwise, the patient may not get better. Okay? So, gram staining is very useful. Now, that's one way of identifying. So one way of identifying bacteria is to see which color they take up, which stain they take up selectively on gram stain and other stains. Another way of identifying bacteria is by their shape. So we can say bacteria are like, little circular or ellipse-like thing. These are cocci bacteria. That's what that's called. That's called cocci. For example, streptococcus. Those are a strep throat causing bacteria. It's a cocci bacteria. They look like this. They don't look like that. So they're cocci bacteria, and they're bacilli. They look like this. They're like longer bacteria. So these are the most common types of bacteria: the cocci shape and the bacillus shape. And then they can be spiral shaped. They're funky looking spiral bacteria. Oh yes. So syphilis, for example, is a spirochete like this. Is a spiral bacteria. So in broad category of bacteria, common ones are cocci shaped or sphere-like shape or rod shaped, or the bacillus. So they're cocci and rod shape or bacillus, and spiral shape or the spirochete. Those are the structural. There are other ones. This is a coccus, and then you can have, this is a bacillus and this is a coccus. Now, is it a combo of the two? There's a coccibacillus, meaning it's not exactly coccus, it's not exactly bacillus, it's like coccibacillus. Yes. And there's a vibrio. Vibrio is like curve shape, like comma shaped bacteria. That's Vibrio cholera, for example. Cholera is like a comma shape, like this kind of bacteria, Vibrio. Okay? So coccus bacteria, bacillus, and the spirochete, the spiral shape, spirillum and spirochete bacteria. This is more spirally than this one, right? So bacteria can be classified based on how they take up stains, and they can be classified as how they look under the microscope. See, then you can use the combination of these two. See, you can say we have a gram-negative spirochete bacteria, or you can say you have a gram-positive bacillus. See, then you can see how you can divide up different bacteria, and you can classify most of the bacteria using simple stain pattern, simple stain that they take up, along with the general shape of the bacteria. So that's the classification of bacteria. Now, how do bacteria reproduce? Now, bacteria reproduce in a different way than eukaryotic cells. Okay, bacteria reproduce primarily by asexual reproduction. Now, asexual reproduction. What is the asexual versus sexual reproduction? See, asexual reproduction implies basically making a clone of the individual, making a clone of the bacteria. So in asexual reproduction, there's no genetic variation from one generation to another generation. There's an asterisk there. Then how come you have different bacteria? Of course, there's some other forces that can result in genetic variations, right? But anyway, the definition of asexual reproduction means there's no genetic variation that occurs because you're kind of making carbon copies of things. And the process of asexual reproduction in bacteria is called binary fission. Fission is to divide, right? Binary fission is to make one into two. So asexual reproduction of the bacteria is called binary fission. Now the difference again between asexual and sexual reproduction is in sexual reproduction there's exchange of genetic material that occurs between two different cells, and the resultant daughter cell is very different from either of the parent cells. So sexual reproduction implies that there's exchange of genetic material between different cells, and daughter cells, the resulting cells, is different from either of the parents. So that's the distinction between asexual reproduction. So asexual reproduction is basically kind of like cloning, duplicating the bacteria. So bacteria, they don't have a nucleus, see? So there's no mitosis or meiosis that occurs in bacteria, because both mitosis and meiosis, what happens? First you form chromosome. Why do you need to form chromosome? You got only like one circular DNA, and then you don't make a spindle fiber and all that stuff, because you don't have chromosomes to divide and stuff like that. So there's no mitosis, there's no meiosis in bacteria. It's a asexual reproduction, kind of simple in terms of what you need to know for exam, so to speak. They divide by binary fission. So what happens? Well, first thing that happens is the DNA has to get replicated, because otherwise how are you going to make two bacteria out of one bacteria? So DNA gets replicated. So what happens is the DNA it's a circular DNA. So what happens is in one area of the circular DNA, you start making a second strand, and you keep doing that, and then in the end you have like two thus B's, so to speak, right? So you have one thus B becomes two thus B's, right? So that's what you do. And then once you have the two copies of DNA, the DNA attaches to the plasma membrane. See, it kind of like hangs itself to the plasma membrane, and then to the opposite side of the bacterium, and then the bacteria grows a little bit and divides into half. So okay, let me say this slowly. So DNA is replicated. Each replicated DNA attaches to the plasma membrane, kind of like hangs on to the plasma membrane, and the cell elongates, right? Cell elongates, and then it cuts into half. It produces two daughter cells. Start. DNA replication. DNA strands separate. And then cross-wall formation is kind of like divided into two. So this is how bacteria reproduce. They reproduce by binary fission, asexual reproduction. Some bacteria reproduce every 20 minutes. They reproduce a lot. So within a matter of time, they come back the next morning, it's like bacterial overflow. Well, there are limiting factors. I mean, they can't just keep dividing unless they have nutrients, right? How are you going to make DNA if there's no nutrient available, no nucleotides available? So if you put a petri dish with the bacteria inside of it, the bacteria is not going to grow, and it's not going to cover up the entire table, because there's no food for the bacteria to grow up on the entire table. It's going to grow until it's used up all the nutrients that are in the petri dish. The petri dish has food inside for bacteria. It has jello is the food for bacteria. Once all the food is gone, the bacteria will stop reproducing. So there are limiting factors of nutrients or poisonous waste, of their own or from their predators, because bacteria have excretions too, and some of those can be damaging to their environment, prohibiting their growth. Now, bacteria, there's some element of bacterial reproduction that's sexual reproduction. So this process is called conjugation. So bacterial sexual reproduction is called conjugation. So what happens is there's some bacteria here. See, there's an F plus cell and an F minus cell. Okay, for example, this bacteria here has a plasmid here. The end result is going to be that both of them are going to have the plasmid. See, so this bacteria has the plasmid. This one doesn't. It's like it's got plasmid envy, so to speak, because and that's what it's called, F minus. Okay. This is F factor. So the plasmid, no plasmid, and the end result will be so. So what's happening here is the first bacteria sharing its plasmid DNA with the other bacteria. Now this bacteria is able to do that. Now this bridge that forms is called the conjugal junction from the pili. The bridge that forms from the pili. Now who forms that bridge? It's this bacteria over here that got the plasmid. Only the bacteria that is F plus, the one that's able to share. So the genes that are needed to make this bridge come from plasmid and from this bacteria. Okay. So anyway, this bacteria makes the bridge, the conjugal bridge. Okay. Basically, it just makes the pili go big until it touches the other one and infuses with the other one. So it makes this bridge, and then this plasmid DNA is duplicated, and while it's being duplicated, the DNA is transferred to the other cell, and then this plasmid remains there, and now this bacteria has the copy of the plasmid. So you form the bridge, the DNA is copied, while it's being copied, is sent to the other cell. Now this cell, called F plus cell, the donor cell is called a factor plus cell, the donor cell, F factor, and then in the end, they both become F plus because they both have the factor. Okay, so this is how bacteria are able to communicate, they share the DNA. This is how antibiotic resistance can, this is one way of antibiotic resistance, can go from one bacteria to another bacteria. So this cartoon picture is showing how this is happening. This is bacteria has a plasmid, F plus plasmid. It's got the pili, that's the nucleide, see? This is called a sex pilus or pilus. And then now here is sharing the entire genome, see? Because the plasmid that has the ability to do this becomes part of the circular DNA, like this. Okay? So this bacteria is sharing its entire DNA to the donor bacteria, like that. Anyway, so that's conjugation or bacterial sexual reproduction. This is a cartoon picture of the same. Now, some bacteria has certain adaptations. Okay, anaerobic bacteria, or possibly the first photosynthetic bacteria, giving off oxygen in the process. Okay, anaerobic bacteria are bacteria that do not require oxygen. See, we cannot live without oxygen. We are dependent on oxygen. There are many bacteria are like that they are absolutely dependent on oxygen. As you can imagine, these bacteria that absolutely need oxygen were not the ancient bacteria. Okay, so but the ancient bacteria, they don't need oxygen, but they may produce oxygen. See, they may produce oxygen. That's how we may have originally gotten the bacteria. So anaerobic bacteria are bacteria that do not need oxygen, okay, but they possibly were the original photosynthetic bacteria giving off oxygen. They don't need it, but they give off the oxygen. So in terms of these type of anaerobic bacteria, in other words, the bacteria that they don't need the other type of bacteria, the bacteria that need oxygen, okay, those are called aerobes. So bacteria can be divided into anaerobes, this is this previous slide, that they don't need oxygen, and then there are bacteria that definitely need oxygen. Okay, the tuberculosis bacteria, which causes lung disease, Mycobacterium tuberculosis, it absolutely loves oxygen and cannot live without oxygen. Okay, so that's why it goes to the lungs, because that's where all the oxygen is. Okay, so Mycobacterium tuberculosis, the tuberculosis bacteria, is not just an aerobe. That means it requires oxygen. It's an obligate aerobe. It means it cannot survive without oxygen. So it requires oxygen for its cellular respiration. So bacteria, they need oxygen to break down their glucose material. So, and obligate aerobes require oxygen for respiration. Obligate anaerobes are bacteria that absolutely cannot have any oxygen around. I mean, they're killed by oxygen. Okay, Treponema pallidum, the syphilis, the STD-causing bacteria, is an anaerobe. That they not only do they not need oxygen, the oxygen is like poison for them. They absolutely do not want any oxygen around. Clostridium botulinum, this is the botulinum, the poisoning, causing bacteria, food poisoning, is also an obligate anaerobe. Okay, and there are bacteria that are neither aerobic or anaerobic. They do fermentation. So bacteria can either, this is another classification form for bacteria. As we suggested, there are different ways of classifying bacteria. One way of how they stain, for example, the gram staining. Second way is their shape, and the third way is whether they require oxygen, whether they do not require oxygen, and how they handle oxygen. So these are three important ways that we talked about how bacteria can be classified. Okay. Another special feature of some bacteria is spore formation. Spore formation is kind of like, an escape pod from a spaceship. That's what that is, escape pod. See, spore is escape pod. Like when things are really bad, certain bacteria, they go into spore formation mode. So they make spores. They hibernate. So spores are hibernating version of the bacteria. For example, one bacteria that we commonly see is Clostridium difficile bacteria, and it's very difficult to treat, sometimes requiring prolonged antibiotic use. And Clostridium difficile is difficult because it makes spores. You give the bacteria antibiotics, it knows there's an antibiotic around, and it makes spores. So this is what happens. So bacteria, what it does is collects its DNA. It collects its DNA into a big lump, and it wraps the DNA into, like, I'm not kidding you. It's like it takes duct tape, right? If you take the DNA, imagine the DNA, you just keep duct taping the DNA into a big ball, right? And the duct tape is the plasma membrane. It just keeps wrapping the plasma membrane around the DNA, and that's the concept of a spore. So, and the rest of it kills everything else, and everything that the bacteria needs to mature and come back after, whenever it wants to come back, is contained in the spore. Okay, so spores can survive for years, for centuries, in fact. Okay, so endospore is a tiny structure with bacterial DNA. It resists drying temperatures, extreme temperatures. It can resist chemicals. It can resist antibiotics. So what we do is sometimes, we trick this bacteria, Clostridium difficile bacteria. We trick it. So we do is, so we give the patient antibiotics, and we taper. So you give the antibiotics for 3 times a day, and then you give it like 2 times a day. And the bacteria, the spores are thinking, hey, the antibiotic concentrations are going down, maybe I can come out. But they come out and they get killed by the bacteria, by the antibiotic. And then you give it once a day, and then for a few days, and then you give the antibiotic once every other day, and you fool these spores, and then you have most patients get better that way. So when conditions improved, and after even sometimes thousands of years, spores can come back. So the spore formation is one way bacteria can go in a hibernation mode. So here's an example was a spore formation. See, cell wall, that's the membrane, nucleoid followed by cell division. All right. So now what's going to happen? Look at this one. Okay. See, it's using the plasma membrane to make it like duct tape and making lots of it. This is a cartoon picture. It's even more so than this. The other one disintegrates. You make more cortex. You make it strengthen it, right? And that's a spore. And the rest of the bacteria dies. Who cares about the rest of the bacteria as long as you have the genetic material and everything that you need? Travel pack. So spore is like a travel pack version of the bacteria. Clostridium botulinum is another one of these spore forming bacteria. This causes botulinum. This is very important. The way you kill endospores is you sterilize the heat under high pressure. Okay, so you heat whatever you want to heat, and you sterilize it, and you store it under high pressure. So this is like canned food, sterilized, acidified. Now that's why, see, if you open up a can like this, you use up whatever's inside, and you don't put it back in the fridge like half used, because that's bad news. The reason they were canned is to prevent formation of these spores But if you use half of a can of stuff or anything that's canned, that's why it says that the can is open, don't buy it, because you know why? Because that defeats the purpose. Because if whatever was canned and stored under high pressure, it was done so because it was sterilized and heated and then placed in there, right? Because all this stuff was boiled. See, this was boiled and stored in high pressure. If you open it and then, if you're not careful about it, right, and then people can get infections, particularly in canned food stuff. Okay, botulism toxin this bacteria produces a nerve toxin. It produces nerve toxin, and that can be very bad because everything in your body is controlled by nerves. I'll tell you some important nerves in your body. Well, let's think about some important nerves in your body. Well, how about the nerves that control the movement of your eyes? If those are messed up, you start seeing double vision. Okay, how about, can you tell me another important nerve? Well, how about the nerve that goes to the diaphragm? Controls your breathing. If that one's paralyzed, that's bad, right? How about the nerves that go to your tongue? So yeah, that's what botulism does. So this bacteria makes spores. That's why you have to be very, very careful about canned foods and stuff. So somebody who doesn't, the other thing about it, is stuff that's heated and placed in aluminum foil type things. Okay, so just be careful. And one thing I always tell my family is to never store, like if you have meat and then you have veggies, you won't mix the meat and veggies in the fridge, because that's like basically nutrients for bacteria that want to cause trouble. Okay. So don't store salad and meat in overnight in the fridge. Just eat the salad and don't put it by the meat. So just be careful with botulism. So muscles cancer. That's that. Bacillus anthrax is another spore-producing bacteria. And anthrax is, of course, is so dangerous that it can be very, very dangerous, as you know. Okay, that's another spore-producing bacteria. Now, anthrax, if a very small amount of it, can cause severe lung disease. See. Now, so we talk about bacteria, all aspects of bacteria. We talked about bacterial structures. We talked about how we identify bacteria. We talked about some of the special features of bacteria, such as spore formation, and how that's very useful information, protecting and keeping yourself healthy. Bacteria are not all that evil. In fact, life as we know it on the planet Earth depends on the bacteria and what they do for us, specifically nitrogen fixation. See, amino acids have a nitrogen group. There's amine group in the end. We know all proteins have amine group, and the nitrogenous bases are nucleotides. There's a lot of nitrogen in organic matter. Where did it all come from? Well, it came from the air, the nitrogen that's in the air. But the nitrogen that's in the air, we cannot pick, and you cannot use it in its form. In other words, we can't breathe nitrogen from the air and use it to make amino acids and nucleic acid. We can't do that. We don't have the enzymes and stuff to do it. So, but we need the nitrogen. So how do we get it? Well, thanks to the bacteria, your friendly neighborhood bacteria in the soil, they do nitrogen fixing for us. So they take the ambient nitrogen in the N2 form, and the bacteria, they have enzymes and stuff. They change it into other organic matter, such as ammonium over here, ammonia, NH3, ammonium, NH4, and we need this, right? And then we need the nitrate form, so they convert this into the nitrate, and then we take this, and plants take this up, and we take it from plants. Plants take this up to make to incorporate that nitrogen into organic compounds. Everything that you have, everything that has nitrogen. So the nitrogen that is in organic compounds come from the air, but first it has to be metabolized by the bacteria. So we need to thank bacteria in this sense, because without them fixing the nitrogen into usable forms by living things would not exist. Now, when plants die, what happens to all of the nitrogen that they took up, right? Well, thanks to bacteria again, they do the reverse process and they free up that nitrogen back into the air. So some bacteria, the bacteria that denitrifying bacteria, the one that eat up plants, they actually are also able to reverse the process. So bacteria are able to fix nitrogen, and bacteria are able to digest the nitrogen that's in living matter and free it back. This is part of the nitrogen cycle. So bacteria are not all that bad. They're one of the primary decomposers in the environment. So fungi and bacteria are the primary decomposers in the environment. Okay, don't worry, we're almost there. Bacterial diseases. Most bacteria are harmless, and in fact many of them are helpful. But when they come to cause disease, they can cause disease in many different ways. To cause disease, they have to first enter the human body. Okay? How do they enter? Well, any which way they can. They can come to the skin if there's a break in the skin. They can infect the skin follicles, the hair follicles. If there's a cut in the skin, they can enter through there. They can enter through the nose and the sinuses. They can enter through the mouth, and they can go into the lungs, right? Or bacteria can be ingested and cause gastroenteritis, right? So these you have to have a portal of entry for bacteria to enter. Okay? So how do they cause disease? Well, anything that interferes with the normal functioning of the structure, okay, or by causing inflammation, or by producing toxin. So if the bacteria, for example, goes into the lungs, our body's immune system attacks the bacteria and causes a lot of inflammation, even if the bacteria will not produce any toxins. And the inflammation that's caused it's like fires in the whole neighborhood in the lung, and it damages the lung, interferes with normal functioning of the lung. So even if the bacteria doesn't produce any toxins, the presence of bacteria elicits an inflammatory response that causes the disease process. Some bacteria, as Clostridium botulinum, Clostridium difficile that we talked about, they produce toxins. Cholera produces a toxin, and the cholera toxin produces attaches to the intestinal cell and basically turns up the faucet, so to speak, so the whole body starts squirting water into the intestines. That's how people die from cholera, because they get dehydrated, because all the water leaks out of these cells, because the toxin causes the leakage of water from the intestinal cell. So it's a toxin producing bacteria. It's not inflammation that causes problems in cholera. It is in fact the toxin. So bacteria can cause disease by either just simple presence and inflammatory response that they elicit, or by producing toxins. This is tuberculosis. Yucky. Normal lung and all this inflammation. Tuberculosis is nasty disease. Bacteria actually eat up the lungs. Mother Teresa, she said this interesting thing. I like this one. The biggest disease today is not leprosy or tuberculosis, but rather the feeling of being unwanted. Yes. So, this is some profound truth to this. I think it's like, I have told you, it's more important to have purpose in life, right? You have to live for meaning. Meaning is always more important than happiness, because if you have meaning, that's your surest way of being content and happy. So strive for meaning, inshallah. That's what I try to do anyways. The story of penicillin is really interesting, because Alexander Fleming over here, he discovered the penicillin. He discovered what would eventually be isolated as penicillin. See, he tried very hard to isolate penicillin. What happened one day, he was growing this Staphylococcus aureus bacteria, and then he was trying to grow this, and then he noticed there was fungus growing. This fungus is Penicillium notatum. Okay? Penicillin comes, at least initially here, comes from a fungus like this. So he saw accidentally in this petri dish, he wanted to grow this bacteria, but he saw this fungus growing. He said, oh my God, I just wasted the petri dish. I've got to do this over again. But he noticed something very interesting. The bacteria did not grow in the immediate environment of this fungus. The bacteria did not grow in the immediate environment. Almost like somehow the presence of the fungus was inhibiting the bacteria. It's like the bacteria were afraid of the fungus. Were the bacteria afraid of the fungus? No. He was smart enough to realize that there must be something about fungus that was inhibiting bacterial growth. And what is it about them? It's not their attitude. is something chemical, some chemical aspect of this fungus was inhibiting this bacterial growth, and this would be called penicillin after the name of the fungus. He tried to isolate this, but he actually couldn't do it. But later on in the next generation, people were able to isolate this. Okay, so the fungus, the mold, Penicillium notatum, contaminated the culture, and he observed that some substance that's secreted by the fungus must be inhibiting bacterial growth. Look at this, bacteria everywhere. So somebody took a swab, and they went like this, see, and then there's a penicillin colony. But the penicillin colony inhibited bacterial growth around it. So that's penicillin killing all the bacteria. Pretty fascinating. When life gives you a mole, you make penicillin. So that is it. Bacteria are very important. Of course, I know this is a lot of material, but I hope that you plow through it. And if you want to review materials, this will be very useful information for you in general, inshallah. And until next time, as-salatu was-salamu ala rasulillah walhamdulillahi rabbil alameen.